Thank you, thank you, thank you. And Jennifer, thank you so much for being here. As somebody who's written biographies, people say, who's the nicest one? I said, there's only one I've written about. Jennifer is the intersection of being a good person and a brilliant scientist, and probably the person most defining our future. So thank you with biotech. How did you, when you were a kid, you were in Hilo, Hawaii. I think you had a guidance counselor who once said, girls don't do science. How did you end up thinking, okay, there are women scientists, I can do this?
I think when I think back to my upbringing, my father was a literature professor. Mm-hmm. He gave me a copy of The Double Helix, a book about the discovery of the structure of DNA when I was probably 11 or 12 years old. And that book really showed me that science is a process of discovery, and I was fascinated by that description of how scientists could figure out the mystery of something in biology by doing experiments. And then I learned about Marie Curie's work and was inspired by her story, and together I think those two ideas really spurred me on to thinking about a career in science.
One of the things in James Watson's book, The Double Helix, is he kind of minimizes and dismisses, dismissively a bit, Rosalind Franklin, who did the great photograph that helped Watson and Crick understand the structure, calls her Rosie. Most people, when they read that book, they say, oh, it was dismissive, but when you read the book, what did you think?
When I read the book, I thought, that's ridiculous. Of course she was doing important experiments.
And that a woman could be a scientist.
Yes.
Which you told me at one point that up until then you hardly knew that there were women scientists.
That's true.
Exactly. So from there you decide to go to not be a French teacher, but go into biochemistry, and chemistry mainly. Why did you choose that? That's not a usual path on chemistry.
Well, I think it started with my chemistry teacher in 10th grade. She was a Miss Wong in Hilo. She taught us kids that science is about solving puzzles and not memorizing facts in textbooks. And I thought that was so interesting. And I thought to myself, wouldn't it be amazing to understand the chemistry of life? So that was really the first inkling I had that really what I wanted to do was be working right at that intersection between chemistry and biology.
Now, on the power of books, your father leaves on your bed the Double Helix. I think it was the old vintage paperback copy.
Penguin books.
Penguin, sorry. And if I remember, you thought it was a detective story because it looked like that, right? And then you found out it wasn't, and then you found out it was.
Yep, that's right. I thought it was a detective story of one type, and in fact, when I read the book, I realized it is a detective story. It's just about something very different than I expected.
Tell me, growing up, you're taking hikes and you're seeing things in Hawaii that we kind of have here, too, out in the, which is weird grasses that if you touch them, they curl up. And one of the things I like about you, Leonardo da Vinci, and others is a curiosity about things that we all see every day, but we don't go, oh my God, why is that? And when you touched, I can't remember the name of the grass, and it curled, you just become fixated on how does that happen, right?
Yeah, I think when I was in Hawaii, I was amazed by all the plants and animals that had adapted to that island environment. So you're mentioning sleeping grass. That's one of the organisms that I was fascinated by, but we also had blind spiders that lived in lava tubes, and this was, something that I found myself drawn to, that question of why.
When you get to Berkeley after a while, through a long thing through Harvard, but when you're there, you weren't doing RNA CRISPR type stuff. I mean, it was not yet a full field. How did you end up starting to study that? I mean, who called you and said, let's do something?
Well, this is the great thing about doing research, is that ideas come out of all sorts of directions. So in our case with CRISPR, the first indication that there was something very interesting going on in bacteria known as CRISPR systems, was the work of Jill Banfield, who studies bacteria in their native environment. And I think she was really one of the first people that noticed that bacteria can acquire immunity to viruses that infect them in real time. And she wondered how that works and why, and she had a hypothesis that involved molecules of RNA, which are the chemical cousins of DNA. She Googled, who at Berkeley works on RNA. My name popped up, and she called me, and that's literally how we got together.
And this is why great research university is so good, because you have a lot of people together, and somebody says, okay, I need to know about this molecule, and you form a partnership, right?
That's right, collaboration.
And one of the problems nowadays is so much of that is funding of basic research, not applied research, just funding for curiosity's sake, that the federal government under Vannevar Bush starting in 1945 made part of what we as a nation do, which is curiosity-driven basic science. So when Jillian Banfield calls you up and says, I got this molecule, I know you're interested in it, but maybe it has something to do with the sequences in bacteria, were you all thinking of an applied application, or were you just basic research?
Oh, certainly not. It was pure, curiosity-driven science.
And what was it you were trying to figure out?
Well, the question at that time for me was, well, first of all, I guess I was amazed that bacteria would have an adaptive immune system. We're all familiar with our own bodies working that way, but nobody had any inkling that bacteria could do something like that. So I was fascinated by that possibility, and also by the role of molecules of RNA, which are thought to be some of the most ancient molecules on our planet, and perhaps even the source of life on Earth.
Well, source of life. So we're all trying to figure out, how did life begin? You had a great, Tom Cech was his name, right? Or who did the origins of life and figured the RNA world thesis that you worked on?
Well, Tom Cech was one person who I worked with in the past, but also my graduate advisor, Jack Shostak, was very interested in this question. Yeah.
And he said, ask big questions, right?
He did.
And what was the big question?
Well, the big question was, where did life come from, and can we study it in the lab?
Mm-hmm. And why does it...
The answer is yes.
But you have to do a chicken and egg riddle to get there, right? Which is, we thought it was DNA that becomes the code for replicating species, but you can't have... How did you figure out that chicken and egg thing?
Well, the fundamental question is, DNA is the code of life, yet it is replicated by proteins that are encoded by DNA. So it sort of sets up this conundrum of, which came first. And some scientists think that, in fact, neither one. It was really RNA molecules that might have had the ability to both encode information, which they do, but also copy information originally, and that union of activities in one molecule could have given rise to early life.
Yeah, I mean, the main thing is to be able to replicate itself, which I guess is what distinguishes a rock from life.
Right.
Yeah. And so one of the things I say in the book, but you've pushed back on, but I'll let you do it again, is when you were doing that, say in the 1990s approximately, all the men, including dear Francis Collins, who I think I see here, and others, they're doing the Human Genome Project. Eric Lander, anyway, it's a very alpha male thing to figure out DNA and to sequence it by 2000. And yet, if I look, there were almost no women on that project, but women like you, Jillian Banfield, Emmanuelle Charpentier, all are focusing on the sibling or cousin molecule, RNA. Why is that, and was that a gender thing or just happenstance?
No, happenstance.
Okay. But you played soccer and you said you always knew to run where the ball was going, not where the ball was. And I figured that was part of what got you into RNA.
I think what got me into RNA, just frankly, was just curiosity and this question about its role possibly in evolution that I found so fascinating, also in the CRISPR pathway.
Well, now that we're talking about CRISPR, I'll start to give it, which means it's clustered repeated sequences that are in the bacteria.
You almost got it right.
Yeah, well, I'm not going to do clustered, repeated, interspersed, palindromic. By the way, it was whatever it's called, where somebody comes up with a name and then tries to come up with the words that will spell the name. Wasn't it the truth? That he said, okay, I'm going to call it CRISPR, and then he had to figure out what would the CRI.
Well, he wanted a nice acronym. So.
Yeah, it was a nice acronym. It's called a backronym or something, where you go backwards to get the acronym. But what it is repeated sequences in the genetics of a bacteria. Explain why bacteria would waste a lot of time repeating sequences.
Well, what's interesting is that it's really as you said, it's a series of repeated sequences of DNA. So you probably all know that DNA is a 4-letter code, and it is spelling out all kinds of molecular information that are required for cells to function. But how do cells mark a particular set of sequences so they know what to do with them? And this is what happens in CRISPR sequences, is that there's a repetitive region in the DNA that tells the cell, this part is special, this is where I'm storing information about viruses that are infecting me over time, so it creates a genetic vaccination card.
It's a little bit like mug shots. They say, hey, this one attacked me before.
Exactly.
And we didn't quite know we might need that as a human species while you were doing it, right? I mean, that we were going to be hit by viruses that way.
Well, we always get hit by viruses, of course, but humans don't have a CRISPR system. They do immunity differently, but in bacteria, this is a very effective way in real time for cells to acquire immunity to viruses and then use it to protect themselves.
Well, they've been at it longer than we have, meaning bacteria have been fighting viruses for 4 billion years or so?
Roughly,
right?
yeah.
And so is this an evolutionary thing that the smart bacteria figured out?
Yeah.
Mm-hmm. And so Jillian Banfield calls you up. Take the story from there.
Yeah, she called me on the phone. This was in the days before we were all, texting each other.
Group chats, we call it now.
Yeah. Yeah. And we met at the Free Speech Movement Cafe at Berkeley, kind of a quintessential place. And she arrived with a big, stack of papers, and she said, Jennifer, we've just noticed something fascinating in these bacterial genomic sequences, and we don't know what it means. But she showed me these signatures of repetitive DNA elements that flanked unique sequences that came from viruses. And so the question was why? Why would bacteria be storing little pieces of viral DNA in their genome? That was the question. And she was so passionate and so excited about this that I couldn't help but, be drawn to it.
And how did that start leading to a gene editing tool?
Well, that led to a whole project that initiated in our lab biochemically to figure out how these sequences might be protecting bugs. And what we figured out, and this is the royal we, with other people working in the field as well, is that these CRISPR sequences encode molecules of RNA that provide the molecular zip codes that tell proteins that are also part of the CRISPR pathway where to go and what to cut. And so what they do in these cells is they cut up viral DNA that gets into the cell and prevent it from causing an infection.
So these are proteins, sometimes called enzymes in this case, right, that know how to cut. They're just like scissors, but they're made of molecules, right?
Right.
And so you see it cuts DNA. When does it occur to you, oh, wait, if I can cut and paste DNA, I can edit genes?
Well, Walter, I remember this morning sitting in my office in Berkeley when Martin Yinek, who was the scientist in Berkeley working on this project, came into my office and he said, Jennifer, we figured out that this protein called CRISPR-Cas9 is an RNA-guided enzyme that has the ability to recognize viral DNA that matches the little letter sequence in these RNA molecules and make a double-stranded DNA cut, just like you would cut a rope. And when we looked at the data, we realized that we had in our hands the knowledge of how to reprogram these Cas9 proteins so they would cut DNA where we wanted. And if one could do that, you could trigger DNA repair in other cell types like plant or animal or even human cells to make targeted changes in the genome. And this is, was really the synthesis of a lot of other scientists' work in the field, but realizing, putting all of those pieces together with our knowledge of this CRISPR enzyme made us recognize that we were probably sitting on a very powerful technology.
And what did you think at first that this ability to edit DNA in humans, would be good for?
All kinds of things. I mean, people were already able to use earlier forms of genome engineering to make targeted changes in DNA. So imagine that you could, perturb a gene and understand its function, or maybe perturb a whole set of genes. But even beyond that, what if you could actually change a DNA sequence to correct a disease-causing mutation? I think that was really one of the things that first attracted our attention.
Well, give us, I think, what may be the simplest, which is sickle cell anemia, is just a one-letter mess-up, right, a typo. And your technology has done what with that?
Right, well, this is a disease that's been understood for a long time at the genetic level, but it was impossible to cure it, certainly, and not trivial to treat. And of course, if you know anyone with sickle cell disease, you know that it's a terrible disorder that causes repetitive cycles of crisis where patients have to get blood transfusions. That's really the only way they can be treated up until CRISPR came along. But with CRISPR, it's now possible to override that mutation and give patients back a normal blood supply, which means that they're free of these repetitive crises.
How big of a deal is it to cross the line between doing that in a patient and doing that in the inheritable genetics of a patient so that the children and grandchildren will have been edited?
Well, now you're talking about something that I think is really interesting and fundamental about a technology like CRISPR, which is that it enables making targeted changes in the DNA of an individual, as is being done currently for sickle cell treatments. But in principle, it could also be done in embryos, where it creates a change in DNA that can be passed on to future generations. We call that a heritable change. And to me, that's really kind of in a different category.
One guy who has done it in China, He Zhuangqi, who visited some of your seminars out at Cold Spring Harbor, he's the only person who has crossed that line, right? Or the only person we know.
That we know of.
And even China punished him. It's like, okay, because you helped get a consensus around the world, let's not cross the line of inherited or heritable gene editing. Do you think that should, tell me about that line and how it can hold?
Well, I think the current state of the field, and this is true even now, is that there's very little information about how genome editing would actually work in embryos, to the point where it's really not, I think, technically safe to use it in that setting. So many scientists think that it's irresponsible to proceed with that kind of an application until we have really vetted the technology and also determined under what circumstances and which conditions would it really be the right way to proceed.
But in some ways that begs the larger moral question, which is, suppose it was something you could technically do, as you can easily imagine, that in 5 or 10 years we'll be able to do it without mistakes or hallucinations, as we call them in AI. Is there just a huge moral question? Well, actually, let me tell you a story. When I was doing it, the book about you and gene editing, there was a young kid, I'm blanking on his name, his picture is in the book, and he loved playing basketball, except for when he crumpled over on the floor because he had sickle cell.
David Sanchez.
David Sanchez. And so he's working in the Bay Area being treated, and one of them says, we'll be able to edit this out someday. In fact, we'll edit it out so that neither you nor your children or anybody, grandchildren, will ever have it. And he said, that's great. And then he comes back and he says, well, wait, shouldn't that be my child's decision? They said, well, didn't you hate to have it? And he said, yeah, but there were things I learned, including resilience and getting up off the floor when I fell down. that maybe we should be careful about editing for future generations. So that seems to me the larger issue than can we technically get it right?
Yeah, I agree. It's a really moving section in the story of David. You know, David, his story was told in the film Human Nature, documentary, and it's a fascinating reflection that he has, even as a young boy, about what is it that truly makes us individuals, makes us who we are. And I think he, you know, really appreciated the fact that, you know, his disease had, you know, it was a terrible, terrible disease, and I'm sure he wouldn't wish it on anyone, but he also reflected that it had helped to shape him as a person, and that he would be different without it. So it, you know, it raises an interesting challenge.
Yeah, but we've always had those challenges. I mean, I'm not sure Jonas Salk or Sabin, people would say, wait, if Franklin Roosevelt hadn't gotten polio, he would have remained an Upper East Side playboy, so let's keep polio.
Interesting example, yeah.
It's from Doris Kearns Goodwin, who's one of my next interviews here, but because she did the polio's effect on Franklin Roosevelt. But do you, let's go back to sickle cell. You can change the letter so the cells aren't sickled. In theory, I know we haven't done it in practice, you could probably change it so that the cells carry more oxygen rather than less, more oxygen than on average, and you could edit so that your children would win the Olympics or be sprinters. Is that morally acceptable?
Well, let's just first point out that you could only do those things if you knew which genes to edit and which mutations to make. But, you know, if we say for the sake of argument, suppose you did know those things. I think you're right. It brings up a very important question we all have to grapple with, because I think this technology will be capable of making those kinds of changes in embryos in the probably not distant future. And we have to decide, are we okay with that?
And one of the things I admired about Jennifer and why I wanted to pick her as the subject, I didn't know you were going to win the Nobel Prize, which helped the book a bit, but was that once you do this and you have this tool, you start worrying about these questions. And in the history of science, we have so many examples. Oppenheimer, the movie, is somewhat about it, which is the Prometheus problem, and that we have snatched a technology from the gods and who knows what we're going to do with it. And early on in biotechnology, I'll call it, a bioengineering, there was a group called the Asilomar group in California that said, okay, this could be dangerous. And they met a few times and said, we don't want government to regulate it. We don't want to let the genie out of the bottle. And they did that process. And what impressed me about you is that when you got this discovery and technology right, you almost replicated, right, the Asilomar process. So you said, okay, let's try it again for this.
Well, I really admired that scientists in the 70s had grappled with these sort of fundamental ethical questions about biotechnology. In that case, they were looking at examples of modification of bugs that live, you know, bacteria that live in the human body, and wondering whether there could be health risks to making those kinds of modifications that had become possible. So we actually contacted Paul Berg and David Baltimore, two of the scientists who were some of the leaders of those original group discussions in the 1970s, and they came to an early conference we had in, I think it was 2014, to discuss the ethics of CRISPR and how we should think about it and how we should proceed as a scientific community.
And how did you enlist it? Because if it was only U.S. scientists curtailing it, then we'd fall behind. It's like the AI argument. Let's not curtail it here because China will do it. How did you try to make it international?
Well, we reached out to scientists in other countries, including in China. We got the scientific academies involved, and this was, I think, really critical to bringing together a global community of people who could think together about how to proceed.
And what lines have you sketched out or drawn on the use of this technology that have been agreed to, or at least a consensus?
Well, Well, I think one of the real challenges with something like this is that it's, I mean, we didn't say this earlier, but maybe folks here understand this already, but, the thing that's so powerful about CRISPR really is that it's not difficult to use. And so that's meant that it could, take off very quickly as a powerful tool, but the flip side is that it's, it's kind of readily deployable for these other purposes. And in the case of the meeting that we had in California to discuss the, kind of the early days of CRISPR and the ethics of it, we really wanted to make sure that scientists would get on board with the, kind of the responsibility that we thought we all had. And so our approach has been to publish articles about this, to get the World Health Organization involved in creating a registry where scientists can be very transparent about work that they're doing, and also to get the scientific journals involved in ensuring that work that gets submitted for publication is reviewed with a lens on ethics.
Are you, how worried are you, or is there a reason to worry about what's happening now in Washington, both with Robert Kennedy Jr. at HHS and the government, in terms of, first of all, let's talk about regulations and science?
Well, it's an interesting thing. I mean, there's kind of two sides to that coin. On the one hand, I think we appreciate the importance of regulations, and especially when we think about approving drugs that are going to be used in us or our kids. We want them to be safe. We want them to be effective, and that's the job of the Food and Drug Administration in the U.S. The flip side is that if you have too many regulations or regulations that don't really make sense, then that can slow down the process. And I think I certainly have seen, both sides of that. So it's a delicate balance. How do you get the regulations right so that they do what you want them to do and protect us, but not impede progress?
And give me some examples where you've taken it from the lab to the bedside in a way by creating commercial companies to develop drugs.
Well, this is the thing. I mean, companies play a very important role in that pipeline. Academic scientists are great at innovation. We love our students coming in with their ideas and having the freedom to pursue things. And that's really, I think, what gave rise to something like CRISPR. But when it comes to expanding on an idea to the point where it can be globally accessible, academic labs are really not appropriate for that. We don't have the funding to do it. We don't have the resources and the personnel to do it. This is where companies are necessary. So I've long believed that there's a really important partnership between academics and companies, and we have to figure out how to forge that effectively.
And Berkeley is very good at allowing people to commercialize and to start companies with the intellectual property, right? No? Okay.
They're
not as good as Tulane, I mean, but yeah.
Well, yeah. They could be better, but
How?
Yeah. Well, I think there are a number of challenges. As we just said, universities are not set up to be companies, and that means that intrinsically, and I'll just speak for my own institution. I don't know about Tulane. Tulane may be much better at this, but really figuring out how to foster that connection. For example, how do you train scientists that are coming out of academic labs, that are trained as academics, to be good business people? How do you do that? There's no sort of easy answer to that. How do you de-risk an idea so that investors are now willing to put money into it, where they think they're going to get a return? That's also not easy. So, and these are not unique to any one institution, of course, but this was part of the motivation for us to establish the Innovative Genomics Institute 10 years ago, which is a partnership between different campuses of the University of California that's expressly focused on this kind of smooth pipeline between discovery and application.
That's something that's happened in a few places over the past 50, 60 years. I mean, I teach a history of technology course here, which is when MIT and Harvard resisted the commercialization of the things, Stanford University under Frederick Terman, who was the provost, encouraged graduate students, starting with Hewlett and Packard and ending with Larry Page and Sergey Brin, that if you had a good idea, form a company. To what extent do you think that process can be improved?
Well, again, I think that we need to do better. I'm speaking to myself here, really. We need to do better at giving our students the training that they need to be effective in business. And one thing that's very interesting is that, and I see this in my own lab, that people that are coming out of our labs, some of them are very focused on the science, and they want to stay that way. Others want to, take a different lens to it. They are willing to, or maybe even happy to let other people do the actual science. What they really want to do is they want to think about the business model around it. How do you expand it? How do you develop it in ways that will solve real-world problems? And frankly, you need both.
I ask all this because we are at Tulane trying a bio-innovation zone, a Tulane Innovation Institute. All of this is happening now. And when you discovered this, you had 2 or 3 really great graduates. I think Lucas was one of them. And you said, okay, we're going to just call the company Mammoth, and we're going to make T-shirts. And they knew how to form a company. Last time I was coming in from the San Francisco airport, there's a huge building that says Mammoth Pharmaceuticals. So explain how you picked the graduate students and said, you can form a company and I'll be, I guess, scientific advisor.
Well, they kind of are self-selecting. These are often students who recognize that that's their interest and that's what they want to do. And I feel like my job is to help them get there. And I love, working with people in the lab, helping them figure out what they're really good at, and then do more of that. And so in the case that you mentioned with Mammoth Biosciences, that was a wonderful situation where there were 2 graduate students in the lab, Lucas Harrington and Janice Chen, who were working together on a project. They both recognized that there was an opportunity to commercialize it. They wanted to be part of that. And they teamed up with a 3rd student coming out of Stanford, maybe the only history of a Stanford-Berkeley partnership successful.
Like Tulane LSU.
Yeah, right.
Can be done.
Can be done, but rare. And so they started Mammoth Biosciences, and they're going strong.
Getting back to the policy challenges we have now, we talked a little bit about regulation and trying to get the balance right. The more pressing ones, I'll start, well, there are 2 of them I can think of, but I'll start with this. NIH funding being cut radically, and other NSF funding being cut. Is that destroying the seed corn for the future inventions like CRISPR?
It's not a good idea, I mean, thank you.
You're not quite as forceful as Tony Fauci was. He
Well,
used stronger language than not a good idea.
Well, let me expand. So you may appreciate that in the United States, we are a leader around the world right now in science and technology. Why is that? It's because taxpayer money for decades has gone into funding the kind of science that we're talking about here. For curiosity-driven science that is asking questions about how nature works, and then taking those key insights that come out of that kind of work and turning them into applications. Companies aren't going to do that. Why not? It's too risky, right? It's just, the companies are not going to be able to invest in the kind of curiosity-driven science that does provide that pipeline, but does so in a way that is, kind of open-ended. And if we cut that off, I guarantee that we're going to see a big change, not only in this country, but around the world, because right now, the United States really drives the discovery of all of the, not all, but many of the medicines that we take and the kinds of technologies that have had such a huge benefit.
I would think that if you were an enemy of the United States and you wanted to destroy its future, one thing you would be doing is say, they did the internet, they did all these things, AI, all because of these science grants. Even Larry Page and Sergey Brin are on National Science Foundation grants when they were graduate students. And you'd say, let's pull all these away so that China can be doing it. Do you worry that a country like China will end up being in the fore if we keep this path?
Oh, I not only worry about it, it's already happening. I mean, it's...
Oh, wow. Explain, give me some examples.
Well, I think we're already seeing scientists being recruited to other countries. They've been very, very proactive already about reaching out even to people in my lab about job opportunities. We're seeing that some universities in the US are already cutting back on their graduate training programs due to NIH cuts or anticipated cuts. And it's not going to get better unless there's a real change in the approach in Washington.
And one of the related things is this, I'll call it, crackdown on visas and people who are on student visas, sometimes getting arrested. I mean, so having foreign students studying here, that's going to be harder for them. Have you seen any problems with that yet?
Well, you know, science is really international, and it's international not just in the sense that there are people all over the world working on scientific problems, but it's international here in the United States in the sense that we recruit many of our scientists and our trainees from other countries. Why is that? Well, again, it's because the U.S. has been a real magnet for them, right? It's attracted them to come here because of the wonderful opportunities that they have had. And if we stifle that, it's going to be a disaster for them.
Have you seen stories of researchers who now are afraid because they're on visas that they may lose?
Well, sure. I think that's happening all over. And we're seeing, you know, some frightening examples of students even being pulled off the street, which is really shocking.
And does that have a ripple effect even at Berkeley?
Oh, sure. I mean, I think it you know, creates an atmosphere of fear.
Mm-hmm. What would you do to try to make sure we became a magnet for the best around the world?
Became? You mean?
Regain. Make sure we stay a magnet.
Well, I wouldn't be proceeding the way we are currently as a country. I mean, I think we have to be welcoming to people from other countries. We have to be willing to support science with taxpayer funding in ways that have been so valuable in the past. I didn't mention before, Walter, but you know, our very first grant that supported CRISPR research in my lab was actually from the National Science Foundation. Mm-hmm. The NSF supported our work long before anybody appreciated that there was going to be human health value to it. So.
And they did that just out of curiosity.
Thank you.
Yes, thank you.
Yes.
Going back to CRISPR, we talked about sickle cell. There's many other applications. Tell me, in humans first, I know there's for agriculture, climate, and other things, but in humans, how will it be applied, maybe even in cancer research?
Right. Well, you know, I think one of the things that's very interesting about CRISPR is that as the first applications are coming to the fore, we mentioned sickle cell disease, but there are also several therapies for liver diseases that are already in the last sort of third phase of clinical trial testing that are looking very promising. These are all for genetic diseases that are relatively rare in the population. But I think that what we're going to see over the next decade of CRISPR is increasingly this technology being deployed to prevent disease and to cure diseases that affect many people. You mentioned cancer. I think there, you know, we're looking at opportunities with programming the immune system in ways that allow targeted cancer therapies, and also thinking about ways that we can provide preventative changes in DNA that will protect us from disease.
Are you suggesting something akin to a cancer vaccines?
I think that's a possibility, yeah.
And how would that work?
Well, the idea would be to program immune cells in a person so that those cells could find and destroy tumor cells before they form a tumor or before they metastasize.
Amazing.
Yeah.
And explain, it works, let's say, messenger RNA and guide RNA. The guide RNA is what you did for gene editing. Messenger RNA is what we use for the vaccines. But it tends to tell our cell, make this protein or something. What are the implications of that, of saying, okay, let's code our molecules the way we code microchips?
Well, what's interesting about using RNA to do that kind of therapy is that it's a transient thing. That means that it happens briefly. And so with CRISPR, if we were to use mRNA, for example, just as was used in the COVID vaccine, to deliver CRISPR molecules, then you could imagine a short-term production of the genome editing molecules that could make targeted changes and then go away, which is kind of ideal. So then you'd have the protective change made, the editor goes away, and a lasting treatment.
But people looking at the mRNA vaccines, who are the anti-vax people and whatever, and some of them in government now, have been implying that a messenger RNA or some guided thing like that will totally change your DNA and is a permanent thing. How could one get across the fact that no, RNA doesn't even go into the nucleus of the cell if it's building a protein, it just programs the outer. I mean, it's complicated to make people believe that they're not getting reprogrammed.
I think this is where, as you know, we scientists have to do better at explaining our findings. Right now, there's zero evidence that there's any permanent changes that are made with mRNA use. Mm-hmm. So there's just no data that would support that conclusion. Yeah,
you just said something interesting to me, which is we scientists are not good at it. I mean, one of the reasons I wrote this book and others do is, wait, let's explain the. Scientists used to be better at being public intellectuals, from the old days of Carl Sagan and others. What should science, instead of blaming it on the people who don't get it, to what extent are scientists, should they be doing more to communicate?
Oh, it's critical. I think it's incredibly important. I tell my students this regularly, and I'm sure you do too in your class, right? You really we have to be educating students to be not only great at what they do in the lab, but also thinking about how they explain the importance of what they do. I tell my students, I want you to be able to say in one sentence to your grandmother, why you're doing what you do and why it matters.
In one sentence to your grandmother, what are you doing now and why does it matter?
Thank you. Rewriting the code of life to protect us from disease.
And you're doing it to... Good.
I do okay.
And what about rewriting it to protect us from climate change?
Well, I'd like to do that too.
How?
Well, here's the thing. So, CRISPR is a powerful technology in part because it works across all of biology. We know that it works in bacteria, but it also works in humans, as we've been discussing, and it works in plants because, fundamentally, they are all using DNA to encode their properties. And so we realized in thinking about that fact that CRISPR could actually be used to make changes in plants, but also, frankly, in the microbes that support agriculture that will be beneficial in terms of protecting the climate. So I'll give you an example. So cattle are harboring microbes in their gut, in their rumen, that are important for digestion, but they also produce a lot of methane. So methane is one of the most powerful greenhouse gases, and it turns out that when you look at methane produced from animal farming around the world, it's about a third of the global methane that's released around the world. Imagine that we could reprogram those microbes to not produce methane, and in fact to use that energy to make more meat or more milk. Great for farmers, great economically, and the right thing to do for the climate. So that's what we're working on.
And tell me how close you are and how that would happen.
Well, this is where we brought on board a partner, a third partner, campus partner at the Innovative Genomics Institute, University of California, Davis, one of the world's great agricultural universities with experts working on this methane problem in cattle. And they had shown that you could change the cow diet to control methane production. But it was clearly not an affordable or sustainable solution to the problem. So we got together and we said, look, let's take your knowledge of cattle and rumen microbiology and combine it with the CRISPR technology for reprogramming and make changes in the microbiome of cattle that could be permanent and could reduce the release of methane. And that's what we're working on right now.
How do you feel in this current climate, not just the politics in Washington, about saying, all right, we're going to use RNA-guided things to edit the biomes of our cows, et cetera. Do you think there would be a backlash, or are you going to have trouble getting people to understand that? It seems like it would be demonized right away.
Well, I think we have to be proactive. I mean, we have a big public impact team at the Innovative Genomics Institute to work on the communications about this, to explain the technology, to show the data that we have for the technology, and to really invite a partnership. You talked about scientists needing to be better kind of ambassadors, and I think that has to be not through lecturing, it has to be through real partnership with our communities.
I'm going to talk about myself for a second. What is it like? I mean, I had to trail you for a couple of years. I was in your lab all the time, in your hair all the time, so to speak, or rubber gloves trying to learn how to do things. What's it like to have books and other things written about you? You're not an out there person trying to get publicity.
Well, I'm still stunned that it got done. Do you remember, Walter, that you called me? So it's kind of an interesting backstory because Walter and I had met at the Aspen Ideas Festival where we did a chat like this, and a few generations ago now it feels like, and Walter a few months later called me up one day and he said, I'm thinking about writing a book. And I said, oh, that sounds great. You're always writing books. And he said, no, I mean about you. And I said, well, that'll never happen. I couldn't imagine that it would come to pass. But, Walter is very, very, persistent. And one thing led to another. And I think what's been great about the book, Walter, is that I think you did a wonderful job of telling a compelling story. It's it's kind of a bit of a, it could be a tome, but it's not. It's a kind of a page turner, actually. And you did a great job of interviewing a lot of the people who were involved in the story, telling their sides of it, talking about the way that science really works, the way it really gets done, and there's competition, there's collaboration, that both plays into the things that actually happen in the laboratory. So I think it's a great way for people to try to, really understand the science that goes into a new technology that you might read headlines about, but you don't have any idea where it emerged from.
I mean, you have that in history with great, advances in science. The double helix being, whatever you may think of Jim Watson, just a wonderfully written book. I mean, it is colorful, even if it's maybe too colorful at times. Do you see a role at IGI, Berkeley, Tulane, whatever it may be, of just training science communicators? Not people going to be great scientists, but when people ask me, how do I go into journalism, whatever, I say it's a tough time to go into journalism, but pick a particular field, like maybe science. Do you think Berkeley and others should have a science communication programs?
I do. I think that's very important. I also think that it's important to encourage people that we're coming into contact with to pursue those ideas. I mean, I think that one example from my own lab is a scientist named Sam Sternberg, who was a former graduate student. You know Sam. You've interviewed Sam. And when Sam was finishing up his PhD, he was a wonderful scientist, incredibly talented. I asked him, what do you want to do next in your career? And he said, well, you know what? I think I want to write a book. And I said, really? You want to write a book? And he said, yeah, I want to write a book about the work that went into the discovery of CRISPR, because I've lived through it in your lab, and I think it's just an extraordinary story. And so again, I sort of thought, well, that'll probably not happen, but it did. He did.
It's called The Crack in Creation, and you should buy it, right? Or no, you're talking about A Crack in Creation.
Yeah. And so he took a year off from his research, and he spent time holed up, writing this story. And it was a struggle. I mean, writing is tough, and
editing genes is tough. Writing ain't that hard. I've done both.
I think writing is very. I've done both too, and I think writing is very hard. Either way, but
I did edit. I think it was human kidney cells, right?
Yes, you did.
And I was able to edit in her lab these cells so that they would phosphoresce or
they turn
glow
green.
in the dark. As I'm not a scientist, and I thought, okay, I'm now Dr. Frankenstein. And they made sure that we poured large amounts of chlorine and killed it. So it's a type of thing, though, it would be better if labs like yours or here or whatever could say to kids, come in and just go to the bench and
Yeah.
have a pipette and a
Get
test
some
tube.
experience. Maybe, like you said, they don't have to be professional scientists in the future, but understanding a little bit about how science actually works, I think it's very valuable, and then communicating that to people is critical.
Yeah, we have an anti-science movement seeming to happen now, but it also comes at a time when humanists are intimidated by science. You know, science. It was two cultures system that been written about. How important is it to sort of connect the sciences and the humanities?
I think it's very important. Again, for the same reasons. I think they're fundamental ideas that we're all grappling with. How do we use technologies? And we haven't brought up AI yet, but artificial intelligence, I think, is the same kind of thing where it's powerful, it's complicated. Really understanding how these models, like large language models, are actually working is non-trivial. And then to evaluate, what's the safety of these things, what's the appropriate way to regulate them. These are non-trivial things to figure out. And so I just think that it's going to require a better effort between scientists and technologists and the rest of us to work that out.
But I feel that humanists who care about the morality and all, they're going to be left out of the equation if they don't make the effort to learn some of the science. That if you're clueless about the science, it's going to be hard to discuss, should we do heritable gene editing.
And that's why I loved that you asked me if you could come to the lab and work with CRISPR. It was great.
The two great historic advances of our time in science, just like 100 years ago it was the age of electricity and then the digital revolution, we're seeing two revolutions happen at once that I think are going to be the most transformative of the past 500 years. The life sciences revolution, meaning gene editing at the core, and the AI revolution, meaning artificial intelligence. We saw the Nobel Prize this year being awarded both in physics and in chemistry to AI because that combination. Tell me what happens in your lab, in your work, and in your thought when you combine the power of the AI revolution to the power of the genetic revolution.
Well, when the work was done that was recognized by the Chemistry Nobel this year, which is a program called AlphaFold that allows prediction of protein three-dimensional structures in a very accurate way, our lab and many, many others began using it almost immediately because it instantly provides a tool that we can use to predict the functions of proteins, how they might interact with other molecules, and that's very valuable. It used to be incredibly time-consuming to work out individual shapes of proteins experimentally, and we still do that, but we don't have to do it nearly to the extent that was required previously. And as a result, it accelerates the pace of science, and we're seeing this more and more with other kinds of AI-driven approaches in technology approaches, is that we can do experiments faster, we can increasingly predict the right experiments to do and not waste time on the others. And I think we're just going to continue to see this acceleration of the pace of discovery. It's very, very exciting, but it's also it's a little bit scary too.
Give me a very specific, concrete way we get our heads around it. Maybe take vaccine, where you use AI to totally, say, handle a huge database that humans could never have coped with and discover something that could be a vaccine.
Well, it means that you can quickly evaluate all the molecules that are being produced by a virus or a bacterium that's infectious and try to figure out what are the ways to neutralize it. So,
and how might it work with cancer or something?
Well, similarly with cancer, same thing. Cancer cells often produce molecules on their surface that are not found on normal tissues. So imagine that you could figure out what those are and what they look like and then how to target them.
One of the problems with CRISPR is that it costs a whole lot. I mean, doing sickle cell, it's millions, so you can't really do it. What is the reason the cost is so high, And what could you do with delivery systems to get that cost down?
Yeah, thanks for bringing that up, because that's a very important point. So right now, there's a drug, Casgevy, that's approved by the FDA, we mentioned earlier, for sickle cell disease. And it's extraordinary. I've met one of the patients who was treated in the first trial using that therapy, and it's completely changed her life in a very positive way. So why isn't everybody with sickle cell disease able to get this if they want it? And the reason, at least in part, is the cost. So it's about $2 million a patient right now for this therapeutic. Yeah, not good, not good. And why is that? Well, it's again, in large part, it's for technical reasons. It's because it's not easy to get those genome editors into the cells that need editing, namely the cells in the bone marrow that are the source of our blood supply in our bodies. So imagine that you had a way to do that kind of targeted delivery into blood stem cells in the bone marrow by a simple injection, or even maybe someday it's a pill that somebody could take. That would be incredibly valuable. It would change the whole field, and it would also make it possible to use CRISPR for lots of other types of diseases. So that's really one of the core mission goals of the IGI, is to figure out how to change the technology around genome editing delivery so those kinds of applications become possible.
So, final question. You're at Pomona College. You're thinking of being a French teacher maybe, but you're also holding the chemistry things, and it's kind of fascinating you, and you figure out a path that takes you to the Nobel Prize. For my students here, what should they be doing that will get them, if not a path to a Nobel, a path to helping our society?
Well, all I can say is when I asked my French teacher about switching my major from chemistry to French, she said, no. Stay with chemistry. So it's probably good advice. But no, I always tell my students, you have to figure out what you're really passionate about and pursue it just sort of doggedly and not be dissuaded by naysayers. You have to be able to identify what you really want to spend your time on and then go after it wholeheartedly. And I really see this over and over in my own lab, is that when students do that, they are successful.
Jennifer Doudna, merci beaucoup.